Hey guys, this is your DNA, RNA, and protein synthesis review for Chapter 10. So in this chapter we talked about two things. We talked about transcription, which is making our RNA molecules from our DNA in the nucleus. That RNA molecule called mRNA goes and is translated during translation into an amino acid chain, which therefore forms our protein. So we are going from DNA to RNA, RNA to amino acid chain, transcription, DNA and RNA, translation, RNA to proteins. So let's talk about the structure of DNA. So DNA was discovered by two scientists. Those two scientists were James Watson and Francis Crick, these two guys over here. Now they weren't the only two that discovered it, but they were the two that were given credit. They were the ones that won the Nobel Prize as well as the ones who wrote the paper. Now, DNA takes the shape of something called a double helix. And a double helix, what that means is it kind of looks like a spiral staircase. So what happens is, if you see here, the DNA kind of twists and turns and spins as it goes up. Now, this double helix structure was obviously first discovered by Francis and Crick, but Franklin was the one who photographed it first. So again, you're looking here, as we can see, spiral staircase, it twists and turns as it goes up. You know, kind of looks something like this. So those chains are kind of turning as it twists up, okay? So that's kind of what we're looking at here as this spiral staircase of DNA made of nucleotides. So DNA, or deoxyribonucleic acid, is the molecule that pretty much supports all life. So DNA, it stores and transmits genetic information. So with this genetic information, this is where our genes are stored, which we have about 30,000 genes. This is where, all of our genetic information can be stored. So every DNA molecule contains a couple things. It contains a sugar group, and that sugar that DNA contains is something called deoxyribo. Okay, so deoxyribose is the sugar. It also contains a phosphate group and a base or nucleotide. So this is what the three parts look like. Now, this sugar molecule, we usually draw in a hexagon, but we have our sugar, we have our nitrogenous base, and we also have our phosphate. So that's kind of the basic structure we see there of our DNA molecule. Now, our DNA consists of two strands of nucleotides joined together by hydrogen bonds. So hydrogen, when we look at that structure of DNA, hydrogen would be these green structures being held. These are what hold the two strands of DNA together, okay? And our long strands of DNA are twisted into a double helix, okay? So this is kind of the basic structure of DNA. We kind of need to know this in order to go forward. So we talked about it a little bit about the bonds that are being held together. So there's two types. These dashed lines right here between the nucleotides, those are hydrogen bonds. So hydrogen bonds are the bonds that hold the two chains together. Now, the bonds that hold the sides together, or the rungs of the ladder, those are covalent bonds. Okay, so the sides are held together and are circled in red by covalent. The two strands are held together by hydrogen bonds. So in DNA, we have what's known as a base pairing rule. And pretty much in DNA, what this base rule is that purines always bond to pyrimidines. Now remember, the difference between the purines and the pyrimidines is the pyrimidines are the ones with the single carbon ring, okay? And the purines are the one with the double carbon nitrogen sugar ring. So to be more specific about this base pairing rule is adenine always bonds to thymine, and cytosine always bonds to guanine. So those are the three different nucleotides that we have in our DNA structure. Adenine binds with thymine, cytosine bonds with guanine. All right? So again, purines always to pyrimidines. Now, what happens in DNA replication is we make what's known as a complementary strand of DNA. And what this means is when we have the two strands, this is when we use the base pairing rule. So for example, I have this strand A, T, C, A, T, G, C, A. So this would be like one strand of my DNA. So to make a complementary strand, right, you just write the complementary base pair, T, A, G, T, A, C, G, T. And really all this is doing is we're doing our base pairing rule. A bonds with T, C bonds with G. All right, so we just made our complementary strand, our double helix strand of DNA. So we also have this rule called Chargaff's rule. And pretty much what this rule states is that in DNA, there is an equal amount of purines and pyrimidines. And to be more specific, we have an equal amount of A to T and an equal amount of C to G. So Chargaff was the one who discovered this rule, and pretty much when he looked at it, he noticed, like you see down here, there's the same percentage of adenine and thymine, same percentage of guanine and cytosine. This holds true especially in humans. Now you'll notice that these numbers are slightly off, and the reason that these numbers are slightly off is because there are other sugar molecules in it. RNA, for example, contains adenine, guanine, and cytosine. So the numbers, they're slightly off, but what the rule always states is there's going to be equal numbers of A's and T's and C's and G's every time we look at an individual's DNA. So what we want to look at here is how DNA is replicated. Now, the first step that we have in DNA replication is a molecule called helicase. So helicase, what that does is it unzips the DNA molecule. So it unzips the DNA molecule. You see here, it comes through and it breaks these hydrogen bonds coming through, and we form what's known as a replication fork. This whole open area right here is what we refer to as the replication fork. And all this is doing is allowing the DNA polymerase to come in and bind to this site. So second thing that happens, once we're unzipped, these molecules called DNA polymerase, okay? DNA polymerase, what that does is it adds complementary nucleotides to each of the original strands. So our polymerase, I'm going to write this up here, often referred to as DNA polymerase, adds complementary nucleotides. So in this case, we're forming what's known as a semi-conservative strand. One is our old strand, and one is a new strand adding that complementary. So after DNA polymerase goes through, it adds all of the new nucleotides. Another molecule comes through called ligase. So DNA ligase, what it does is it seals the two molecules together. In other words, what's happening is we're forming or reforming these hydrogen bonds between and so that the molecule doesn't separate. Again, two strands are formed. One strand has the old set of DNA, one has the new set. So, like I said, after DNA replication, we form what's known as a semi-conservative strand. So, the semi-conservative, again, what this means is we have a new DNA strand or molecule, one strand is old, one strand is new. So when they say semi-conserved, it means that half of the genetic information is from the old DNA molecule, half is from adding the new nucleotides to it. So this kind of leads us to our central dogma. And the central dogma of biology is this DNA, RNA to protein. So our DNA is our genetic molecule, okay? And RNA is made during transcription. So in this case, transcription occurs in the nucleus, and it makes a molecule of mRNA, which is our messenger RNA, because mRNA is able to leave the nucleus, RNA is not. So this RNA molecule then goes to the ribosomes, which are located in the cytoplasm. And this is the site of protein synthesis. So RNA goes to our protein molecules to make amino acids. This is called translation. So proteins are what our body uses mostly in order to pretty much read the code. So DNA to RNA transcription forms in the nucleus. RNA to proteins is translation, which occurs at the ribosomes in the cytoplasm. So that's pretty much our whole central theme that we have in biology. Now, before we get going here, we got to talk about a few different differences between RNA and DNA. So, RNA contains the sugar not deoxyribose, but instead a ribose sugar, and this is what gives it its RNA code. So, again, instead of that deoxyribose sugar, okay? So, RNA also doesn't have the molecule thymine. It has uracil. So in RNA, instead of having AT, we have A bonding with U and our normal C bonding with G. And also RNA is single-stranded while DNA is double. Okay, so there's a couple slight differences that we have here. Again, the big one is the A bonding with U molecule. So we have a couple different types of RNA. So we have our messenger RNA, and our messenger RNA is pretty much, this carries the instructions to make our protein. Okay, so this is the stuff that's made in the nucleus and moves to the ribosomes. Now, the rRNA is part of the structure of a ribosome. All right, so the structure of the ribosome is what, the mRNA comes and attaches to. So over here, we have our messenger RNA. Our ribosomal RNA is what helps in the formation of this. Now, the transfer RNA takes this T shape, and what this is this is what transfers amino acids to the ribosomes to make our protein. So these transfer RNA contain our anticodon. So this 3-code sequence, our codon, 3-base sequence, is what the tRNA recognizes, and the tRNA comes with the proper amino acid to make our amino acid chain. So in transcription, again, we are making our RNA. And what happens here is a molecule, helicase comes in, unzips it, DNA polymerase attaches to it, RNA polymerase attaches to it, because this is an RNA molecule, to the DNA, and an RNA strand is made. Pretty simple. Once it hits that stop codon, the whole sequence is released. So transcription, making RNA. Now translation is a little more complicated, and this is where we're making our proteins. All right? So in translation, we have a couple different steps. So the first step we have is what's known as initiation. Okay? And initiation is where this whole sequence binds to it. And we find our start codon, which is our 3 that kind of initiates the whole process. Our tRNA comes in, binds to that to start it, and as this molecule of RNA is sliding through the ribosome, okay, more tRNA molecules are coming. So when multiple tRNA molecules come with their amino acid, we form what's known as a peptide bond to make this chain continue to grow. So this is initiation. Okay? So elongation is when this process is continuing longer and longer, and our strand of amino acid is growing bigger and bigger. So what happens is tRNA comes in, it binds to it, it transfers its amino acid, and then another transfer RNA goes to the next codon. So as this goes through, this chain continues to form peptide bonds, and the amino acids are continually added. So once there's a certain spot on this chain, let's just say it's this AGA, that codes for stop. Once we reach, the ribosome reaches that stop codon, there's no transfer RNA anticodon for it. So what happens is termination occurs and the whole sequence disassembles. Okay? The disassembly, the newly made polypeptide or amino acid chain releases, the ribosome releases, and the last tRNA releases, and the protein goes where it needs to. So again, this whole process here, transcription, making RNA from our DNA molecule, translation, these molecules of tRNA are coming in and attaching to the RNA or the mRNA at the ribosome. Okay? When these tRNA comes in, it forms a bond and allows our amino acid chain to form. So the last thing we want to talk about here is making a chain of this. So what we're going to show you here is, making an amino acid chain. So the first thing we need to do is make an mRNA strand from the DNA strand below. So remember, there's no U or T's in this. So we're going to go U, C, and I'm just reading this sequence right here and making my mRNA strand. U, U, G, U, A, U, U, A, U, G, G, A, G. Okay? So the next step in this process is going to break this down into codons, or 3 nucleotide sequence. Okay? Now I take each one of these. My first codon is UCU. So I go onto this chart, I start in the middle, U, C, U. So my first one I have is going to be serine. And we usually just write that as a 3-letter sequence. I go to my second codon, U, G, U. So again, that would be cytosine. So CYS is what I would write. Okay? I go to my third one, AUU. A, U would be isosine or isolene. Isoleucine, really close. Okay, my next one, AUG. AUG, okay, would be methiothenium. Okay, and my last one I have would be GAG. And GAG, glutamine. So that would be how I would make my amino acid chain from the chart. Now there's two different charts you can use. I use the wheel one in this one. There's also the chart which has 3 sides. We showed that one in class. So that's my little review video on protein synthesis. Hopefully this guys helps you for the upcoming test. And this is Mr. O'Brien signing off.